Rhizobium is a genus of gram-negative soil bacteria that fix nitrogen. Different species of Rhizobium form an endosymbiotic relationship with the roots of primarily legumes and other flowering plants. These bacteria infect plant roots and induce the formation of root nodules in which nitrogen fixation takes place with the help of enzyme nitrogenase. By nitrogen fixation, atmospheric nitrogen is converted into ammonia, which is easily absorbed by plant roots. Ammonia is shared with host plants in the form of organic nitrogenous compounds such as glutamine. The plant in turn provides bacteria with organic compounds that are made by photosynthesis. Because of its nitrogen fixing ability, Rhizobium is used as a biofertilizer. In addition to fixing atmospheric nitrogen, Rhizobium is also capable of solubilizing phosphate. Hello and welcome to my channel, Sun Biology Class. Friends, today we will discuss about the isolation, characteristics, types, inoculum production and field application of Rhizobium. Before that, I would like to convey my gratitude to Mr. Rasmiranjan Misra, reader in Zoology, Jawaharlal College, Patnagarh, and his students for subscribing my channel. Really, it is a lot to me. Isolation and characterization of Rhizobia involves several key steps. First step is nodule isolation. Second step, surface sterilization. Third step, culture on specific media. And fourth step is morphological and biochemical analysis. First of all, let us know about the isolation and culture of Rhizobia. First step is nodule collection. Rhizobium resides in the soil as well as in the root nodules. However, healthy root nodules from mature plants, preferably legumes, are used for obtaining contamination-free Rhizobia. Healthy root nodules are detached carefully from mature leguminous plants and are then cleaned by using distilled water to remove soil particles. The cleaned root nodules are then surface sterilized by using mercury chloride or sodium hypochlorite or 90% ethanol. The surface sterilized root nodules are then crushed in distilled water and the resulting suspension is then diluted. The diluted suspension containing Rhizobia is then plated on a nutrient medium containing yeast extract mannitol agar and then incubated at 28 to 30 degrees centigrade for 3 to 4 days. As a result of this incubation, there will be numerous rhizobial colonies on the nutrient medium from which suitable colonies are selected for identification and characterization. Identification and characterization of Rhizobium. Both Azotobacter and Rhizobium grow on yeast extract mannitol agar. However, Rhizobium is identified by CREMA test. CREMA stands for Congo red yeast extract mannitol agar. It is a nutrient medium prepared by mixing 2.5 ml of Congo red with 1 liter of yeast extract mannitol agar. Congo red is a dye. This is followed by inoculation of CREMA with bacterial sample and is followed by incubation at 28 to 30 degrees centigrade. Rhizobium will utilize very slowly and will form white, raised and circular colonies. By contrast, Azotobacter will display colony characteristics similar to Rhizobium, but the colony color will similar to that of Congorate. Based on this, rhizobial colonies are selected. After clima test, the identified rhizobial colonies can also be observed microscopically. For microscopic examination, the bacterial sample containing purified rhizobial colonies are stained with carbamazepine. Carbamazepine is a dye that will color polybeta hydroxybutyrate granules present within rhizobium. The identified rhizobial colonies are then used for production of inoculum. Inoculum production involves three main steps. First step is starter culture of rhizobium. Second step is mass cultivation, and third step is preparation of carrier-based inoculum. First step is starter culture. To prepare starter culture of rhizobium, pure rhizobial colonies are transferred to yeast extract mannitol broth. This is followed by incubation on a rotary shaker at 28 to 30 degrees centigrade for 4 days. As a result of this incubation, there will be sufficient number of rhizobial cells in the broth. Next step is mass cultivation of rhizobium. For mass cultivation, large quantity of yeast extract mannitol broth is prepared and then sterilized. The sterilized nutrient broth is transferred to a large production fermenter. The pH of the nutrient broth is adjusted to 6.5 to 7 by using potassium hydroxide or sulfuric acid. After pH adjustment, the nutrient broth present in the production fermenter is inoculated with a small amount of purified rhizobial colonies from the mother culture. This is followed by incubation at 30 to 32 degrees centigrade for 3 to 4 days. As a result of this incubation, rhizobium will grow and will get a large quantity of nutrient broth containing large number of purified rhizobial cells. Next step is preparation of carrier-based inoculum. This step is essential for preparing rhizobium as a biofertilizer for application on the agricultural field. In order to prepare carrier-based inoculum, inert material known as carrier is mixed with harvested broth so that the inoculum will be easy to handle, to pack, to store, to transport and also to use. Substances such as peat, lignite, farmyard manure and charcoal powder are used as carrier. First of all, the carrier is dried under the sun and then powdered. The powder is then screened through a sieve, after which the carrier is autoclaved. In the next step, the carrier is mixed with harvested broth with hand or mechanically to ensure proper mixing. In the next step, the mixture containing broth and the carrier are placed in plastic trays covered with polythene and are left at 22 to 24 degrees centigrade for 2 to 10 days. During this period, rhizobium will multiply. This process is known as curing. Last step is packaging and storage. After curing, the mixture containing carrier and the rhizobial cells are packed in polythene bags. The bags are then stored at constant room temperature so that the rhizobial cells will get established. After this, the polythene bags are stored at low temperature so that the rhizobial cells will remain viable for 6 months. In order to apply rhizobium as a biofertilizer on the field, we have to follow this procedure. First of all, take water in a container to which add about 50 gram of sugar or jaggery. This is followed by boiling for 15 minutes, after which about 200 gram of gum arabic is added and then cool it. Gum arabic is added to make the solution sticky. Now we got the sticker solution to which rhizobial culture is added and then mixed properly. As a result, we got the inoculum slurry. To the inoculum slurry, seeds are added, mixed, dried under shade and keep it covered. By adding seeds to the inoculum slurry, we got seeds that are coated with rhizobial cells which can be sown in the field. Friends, this is all about rhizobium. For more updates, subscribe my YouTube channel, Sam Biology Class.